Welcome back. We are talking about the respiratory system, chapter 22 in our books for Biology 2225 at CNN. Last time we left off at objective number 142, so we're talking about the mechanisms of gas exchange. What is it that drives oxygen into the blood from the alveolar air and carbon dioxide out of the blood into the alveolar air? And to understand this, we have to understand some basic concepts of pressure and concentration of gases. And so just to review some things we've already talked about, we know that in a gas mixture like the atmosphere, the total amount of pressure of the gas mixture is equal to what we call the sum of the partial pressures of the component gases. Excuse me. The partial pressure of each gas can simply be calculated by taking the percent of that gas in the gas mixture and multiplying it by the total pressure. And so at sea level, where atmospheric pressure is about 760 millimeters of mercury, for instance, the partial pressure of oxygen, which we call PO2, would be 20.9%, which is oxygen's typical value in the atmosphere, times the total, 760. And that would be 159. So the partial pressure of oxygen in atmospheric air, we say, is about 159. Notice carbon dioxide, much smaller percentage, and so its partial pressure in the atmosphere is much smaller, coming in at 0.3. Again, these will change as the total pressure changes. So in a place like Albuquerque, where our atmospheric pressure tends to be around 600 and change, about 620 or 30, these numbers would be correspondingly smaller. We talk about partial pressure because, in a sense, if you think about it, partial pressure is not only a measure of the physical pressure that the gas exerts on a surface, but it's also a measure of concentration, since it's proportional to the percent of that gas in the gas mixture. So when air arrives at the alveolus, gases are going to move between the blood and the air. But last time we talked about the problem of how do we know which way gas is going to move when we have two different media. When we have gas in one case in a fluid, in plasma, and in the other case in the gaseous state. Well, we have to have a similar way of estimating the concentration of the gases. We already saw that partial pressure of a gas in a gas mixture is in essence a measure of its concentration, but in solution, in a fluid solution, how do we evaluate the concentration of a gas in solution? Well, we could talk again about percent of the gas and so on, but what's nice is that we can use a consistent terminology and talk about the partial pressure of a gas in solution as well. And this brings us to something called Henry's Law. And remember, what Henry's Law stated was whenever you bring a fluid and a gas in contact with one another, and in this case water of the plasma and the air, say in the alveolus, at a given temperature, for a constant temperature, which we assume we have in the body at a consistent temperature of about 98.6 degrees Fahrenheit, the amount of gas that dissolves in water is determined by two things: its solubility in the water, which we can't alter, that's a constant, and its partial pressure in the air, which is variable. And so ultimately what Henry's Law is telling us is that, in terms of the physiological significance of this, is that the amount of a particular gas that's going to diffuse into solution is essentially proportional to its partial pressure in the gas mixture that it's exposed to. And so we can use partial pressure not only as a measure of the concentration of a gas when it is in the gaseous state as part of a gas mixture like the atmosphere, but we can also use partial pressure as a measure of the concentration of a gas in solution. And so you can have a solution, say a glass of water sitting on a tabletop. That glass of water will have a partial pressure of oxygen, a PO2. A concentration of oxygen in it, because from the atmosphere a certain amount of oxygen will diffuse into solution. The concentration of oxygen in that glass of water will be measured as the PO2 of that water. And if we say that the PO2 of the water is 150, what that means is that glass of water, or the water in the glass, has as much oxygen in it as it's supposed to have if it's been exposed to a gas mixture or a gas with a PO2 of 150. All right, so in other words, when we say that water has a PO2 of 150, we assume it's already been exposed to a gas with a PO2 of 150, and diffusion has occurred. And so that gives us a sense of concentration. So if I have a glass of water with a PO2 of 150, and I expose it to a gas mixture that has a PO2 of 170, now I can predict which way gas is going to move. Oxygen should move from the air, from the gas, into solution, because the gas mixture had a PO2 of 170, the solution had a PO2 of 150, and I know then that the gas has more oxygen in it than the solution does, and I can predict which way diffusion will occur. The oxygen will move into solution, and so on. So we use PO2 not only as a measure of concentration of a gas in the gas itself, in the gas mixture, but also in solution. Now, one thing we have to be careful about is that we cannot compare PO2s of solutions for different gases, or partial pressures, I should say, of gases for different gases. So let me clarify what I mean by that. If I have a solution with a PO2 of 150, and I have a solution with a PO2 with a PCO2 of 70. The tendency is to think that the solution with a PO2 of 150 has a greater concentration of oxygen than the solution with a PCO2 of 70 has a concentration of carbon dioxide, because clearly 150 is more than 70, but that would not be true. I cannot compare partial pressures of solutions for different gases. I can only compare concentrations using partial pressure when I'm working with the same gas. So a solution with a PO2 of 170 or 150 actually has less oxygen in it than a solution with a PCO2 of 70. That solution with a PCO2 of 70 has more carbon dioxide in it than does a solution with a PO2 of 150 has oxygen in it. Why? Because remember, solubility is different for different gases. Because carbon dioxide is many times more soluble in water than is oxygen, I can't compare concentrations using the partial pressure across those two gases. Right? So let's say carbon dioxide is 100 times more soluble in water than is oxygen, which is about what it is. So a solution with a PCO2 of 70, you would think has half as much carbon dioxide in it as does a solution with a PO2 of 140 in terms of oxygen concentration, right? 70 and 140. But because carbon dioxide is about 100 times more soluble, in reality, a PCO2 of 70 is going to represent a lot more carbon dioxide in solution than does a PO2 of 150 in terms of oxygen in solution. So just something to keep in mind when we're trying to estimate which way gases are going to diffuse. We can only compare partial pressures when we're talking about the same gas, oxygen to oxygen, carbon dioxide to carbon dioxide. Fortunately, that's fine for our purposes. There's really no need for us to evaluate whether a solution has more carbon dioxide in it than it has oxygen, or vice versa. In terms of gas exchange, we really are only interested in the oxygen to oxygen gradient and the carbon dioxide to carbon dioxide gradient, because those are the only things that will determine which way those gases will flow. So I know I spent a lot of time on this, but it does tend to be confusing to some students, and I hope that clarifies it just a little bit. So as long as we're talking about a single gas, either oxygen or carbon dioxide, we can use their partial pressures to evaluate which way gases should diffuse. All right, so loading and unloading of gases is the term we use to say when a tissue or the blood picks up or releases carbon dioxide. It's always from the perspective of what you're talking about. So blood can load and unload carbon dioxide. Blood can load and unload oxygen. And at the alveolus, of course, what we want is for the blood to unload carbon dioxide, and we want the blood to load oxygen. In other words, release carbon dioxide and pick up oxygen. And of course, as I've said repeatedly, this is driven by concentration gradients or partial pressures in solution and in the gas. And we don't need any kind of active transport or pumping of gases to accomplish gas exchange. It happens automatically via diffusion, which is a good thing because that saves us then lots of energy that we don't have to expend constantly for gas exchange. As you see here, it takes about 1/4 of a second to reach equilibrium as the gases diffuse, and typically because of relatively low pressure, erythrocytes spend about 3/4 of a second flowing through alveolar capillaries. So that is plenty of time for gas exchange to equilibrate as the red blood cells flows through a alveolar capillary. Even during strenuous exercise, as this points out, it still only takes, or the red blood cells still spend about 3/10 of a second flowing through the capillary, which is still plenty of time for the gases to equilibrate. All right, and again, important here, one gas does not influence the diffusion of another gas. So the concentration gradient for carbon dioxide does not have anything to do with the concentration gradient of oxygen, and so one does not influence the other, and that's why, again, we don't have to worry about comparing the PCO2 to the PO2 because their diffusion has nothing to do with one another. So as long as we just compare PO2 to PO2 and PCO2 to PCO2, we'll be fine in terms of predicting movements of gases. Here we see a visual representation of Henry's law, again simply that the partial pressures of a gas, or in other words the concentration of the gas, will determine its diffusion into solution. And here we see in the normal state, of course, we start with more oxygen in the gas, less oxygen in the fluid, in this case blood, and given enough time, which as we just saw was about 1/4 of a second in the lungs, you will achieve a state of equal concentrations. That's your equilibrium state for diffusion. Now remember, that's important too, that the end result of diffusion is an equilibrium state. We are not able to concentrate oxygen in solution. That would require active transport, which we're not doing here. So that's important because I think a lot of people think that when we breathe in and send oxygen to the alveolus, that all of that oxygen that we breathe in is going to move into the blood. It does not. All we can achieve with diffusion, remember, is equal concentrations between air and blood, or you get to an equilibrium state where there is no concentration gradient anymore. But that always means, of course, that there's going to still be some oxygen molecules in the air, about the same concentration as in the blood. And the same, of course, is true for carbon dioxide, except the gradient is the opposite. Of course, deoxygenated blood coming back to the lungs has more carbon dioxide in it than does the air that we breathe in, and so the net movement of carbon dioxide is going to be out of the blood. It's unloading, right? But again, the final concentration gradient is an equilibrium state. There is no concentration gradient, but we don't eliminate all the carbon dioxide from the blood. We just get to a state of equal concentrations. Okay, so what are some actual numbers that help us understand why these gases move? Well, here we see some numbers. Typically, the PO2 of the air we breathe in and send to the alveolus is about 104. And again, all these numbers are assuming sea level. About 104 is the partial pressure of oxygen in the air that's in the alveolus. But in the blood, the partial pressure of oxygen is only about 40, because remember, this is deoxygenated blood, so we expect it to have a low PO2. So what we see there is that's a favorable gradient, and oxygen is going to move from 104 to 40. And so what that means is oxygen is going to diffuse from the alveolar air into the blood, which is exactly what we want. We want the blood to load with oxygen. If we look at the concentration gradients for carbon dioxide, we see that the PCO2, partial pressure of carbon dioxide in the deoxygenated blood, is 46. The PCO2 of the air that's in the alveolus is about 40. And so once again, we see there's a concentration gradient, and carbon dioxide should diffuse from 46 to 40, meaning carbon dioxide should diffuse from the blood into the air. And that, of course, would be unloading. Again, doing exactly what we want it to do. Now, there's a couple of things to think about here. Number one is that these numbers, like 104 and 46, aren't even close to each other. Again, it doesn't matter. It doesn't matter that PO2 is 104 and PCO2 is 46 because we don't care about a gradient between oxygen and carbon dioxide. Those are two different gases, two different solubilities, and they do not affect each other's diffusion rates. So it absolutely doesn't matter what these numbers are compared to these numbers. Just totally irrelevant. Don't even worry about that. One thing I do think is interesting, though, is if you go back to the slide, if you think back to the slide of partial pressure of oxygen and carbon dioxide in the atmosphere, at sea level, the partial pressure of oxygen was supposed to be about 159 in the atmosphere. And the partial pressure of carbon dioxide in atmospheric air was very small, less than 1. So why then are the PO2 and PCO2 of the air in the alveolus not the same as in the atmosphere? Well, again, this is something we touched on last time. Every time you breathe in, the air you breathe in is going to mix with air that's already in your lungs. Because remember, during normal breathing, you don't completely collapse and empty your lungs with every breath. In normal breathing, you still have expiratory reserve volume and residual volume left in your lungs. That's sitting there and not being expired. So when you take in a mere, say, typical 500 milliliters of air, tidal volume, it mixes with the 1,200 or so milliliters of residual volume and the 1,300 milliliters or so of expiratory reserve volume. Well, those gases, that amount of air that was remaining in the system has given up some of its oxygen already and picked up some CO2 from the blood. And so we're mixing that fresh air with air that's already been in the system for a while, and that creates, as you might guess, a lower PO2 and a higher PCO2 than atmospheric air. And that's the primary reason why these numbers are not the same as atmospheric PO2s and PCO2s, because of the mixing of the air you breathe in with the air that's already in the system. Okay, so that brings us up to about objective, again, 143, 144. We're in the neighborhood of the objectives that we left off with last time. All right, here's a nice visual representation of what's going on with the air that with the partial pressures. I like this diagram because it does help people visualize everything we've been talking about. So remember, the air that's in the alveolus has a PO2 of about 104 and a PCO2 of about 40. Again, that's different from the inspired air, which I referenced just recently, PO2 of 159 and PCO2 of less than 1.3. And again, the reason these two numbers are not the same, as I just said, is because of mixing of the inspired air with the residual air that's already in the system. But again, this is still fine because notice the deoxygenated blood, as we just said, coming in has a PO2 of 40 and a PCO2 of 46. So that's the favorable concentration gradient that we're interested in. PO2 of 104, PO2 of 40, oxygen is going to move into the blood. PCO2 of 46, PCO2 of 40, CO2 is going to move out of the blood into the air. And once we reach equilibrium, we're going to have what we call oxygenated blood. What are the numbers for typical oxygenated blood? Well, typical oxygenated blood has a PO2 of 95. Notice that's going to be less than 104 because we've pulled some oxygen out of the air. And of course, it's more than PO2 more than 40, which would be deoxygenated blood, somewhere in between, which the equilibrium state usually is, we end up usually around a PO2 of 95. Again, this is the concentration of oxygen that's dissolved in the blood plasma. At this point, we have not even mentioned hemoglobin yet. So these numbers have nothing to do, at least not yet, with hemoglobin. They do have something to do with hemoglobin, but for the moment, we haven't really talked about hemoglobin, at least not yet. All right, and what about the PCO2? The PCO2 of oxygenated blood is about 40. And again, it's the result of an equilibration between a PCO2 of 46 and a PCO2 of 40. Why isn't the number somewhere between 46 and 40? That's a little bit of a complicated story that we may not be able to touch on. We might just touch on it briefly when we talk about carbon dioxide transport. But for now, we just have to accept that that is the PCO2 of oxygenated blood. And again, notice it is less than the PCO2 of deoxygenated blood, which is what we expect. All right, so we send this oxygenated blood out to the tissues, and notice we also have to perform gas exchange out in the system as well, out in the systemic capillaries. But out in the systemic capillaries, we want the movement of the gases to be opposite. We want oxygen to move out. In other words, we want the blood to unload the oxygen, and we want carbon dioxide to be picked up. We want the blood to load carbon dioxide. And so you'll notice that what we see here is that in the tissues, generally speaking, we have a PO2 of less than 40. I know this says about 40, but in reality, the PO2 is less than 40. And the PCO2 is generally a little bit more than 46. So if I could magically change this, I would turn this to a PO2 of less than 40 and a PCO2 greater than 46. So what's going to happen? Again, we're going to have a concentration gradient established. So if the PO2 of the oxygenated blood is 95 and the PO2 of the tissues is less than 40, again, we have a concentration gradient. Oxygen is going to move from the blood into the tissues, which is exactly what we want. If we look at the PCO2s, we're going to have a PCO2 of the oxygenated blood of 40, a PCO2 in the tissues of greater than 46. And so again, a concentration gradient, CO2 is going to move from 46 to 40, meaning from the tissues into the blood, and that's what we want. We want the loading. Something to appreciate about these numbers here is that they're variable. These numbers will vary quite a bit based on the metabolic activity of the tissue, the degree of blood flow to a particular area, how much perfusion a tissue is getting, whether you're exercising or not, whether you have a fever or not. All kinds of variables can affect the PO2 and the PCO2 of tissues. So that's why we don't have an equal sign really here. We just sort of generally think of these numbers as being less than 40 and greater than 46, depending. All right, so what happens after equilibration? Again, O2 moves out, CO2 moves in, and we end up back where we started, which is with deoxygenated blood. That typically, again, has these numbers of 40 and 46, and we start the whole process over again. So that's how diffusion drives gas exchange between air in the lungs and the blood, and also between the blood and the tissues in the systemic circuit. All right, and here we see again that the amount of diffusion of a gas is dependent on the pressure, partial pressure gradient, which can be thought of again as a concentration gradient. And so here we have an increase in the pressure gradient of O2, right? And what we see essentially is that the greater the gradient, the more gas exchange is going to take place. It's not really a terribly important graph, but that's what it's trying to show, is the greater the gradient, the more gases will diffuse. All right, now again, as I said earlier, remember that we can't equilibrate the numbers. 104, PO2 of 104 and a PCO2 of 46 or anything like that. It doesn't matter. So you may think that more oxygen moves because the difference in the numbers between 104, say, and 40 is greater than the difference between 46 and 40, the numbers for CO2. But again, remember, carbon dioxide is way more soluble in fluid than in water than is oxygen. I said 100 times earlier, it's actually 20 times, so I apologize for misspeaking. But that difference in solubility means that a gradient in PCO2 of 46 to 40 is not necessarily less than a gradient of in PO2 of 104 to 40. So the take-home message of all this is about the same amounts of oxygen and carbon dioxide are exchanged, even though their numbers, their partial pressures, are very different, and the gradients appear to be different. The greater solubility of CO2, in other words, offsets the lower apparent gradient for that gas compared to oxygen. So take-home message, about the same amount of gases diffuse during gas exchange, the same amount of CO2 as for oxygen. And I do want to remind you again that temperature does affect the solubility of all gases in solution. And again, since our body temperatures are relatively constant, we don't think much about temperature, but ecologically, of course, temperature does play a role. And the basic relationship is that the higher the temperature, the less gas generally can dissolve in solution. And so the ecological significance of this is that there is such a thing known as thermal pollution. You see it around nuclear power plants and things like that, where you have cooling towers that take in water from a river or a lake, and they use the water to cool down the reactor, and then they put that warm water back into the lake or stream. It's not necessarily radioactive water, but it is warm water, and that will tend to increase, at least locally, the temperature of the lake or the stream, and that increase in temperature affects how much oxygen can remain dissolved in the water, and that creates a real problem for the aquatic organisms that depend on that oxygen, like the fish and the bivalves and the snails and things like that. And so you can get some die-off, some loss of species as a result of thermal pollution. But again, for us within our bodies, because the temperature is relatively constant, then we don't have to worry too much. Another thing that affects the rate of diffusion is the thickness of the respiratory membrane. Remember, the respiratory membrane is the two squamous cells, one for the alveolus and one for the capillary, and a shared basement membrane between them. And typically, it's only about half a micrometer thick, extremely thin. And so that's by design. The body's trying to create a very thin membrane to facilitate diffusion. However, there are some things that can affect the thickness of the respiratory membrane. Pneumonia and some other diseases can cause a swelling and buildup of fluid, which ultimately can cause thickening of the respiratory membrane as fluid builds up in the basement membrane between the cells. And of course, as the membrane thickens, that increases the distance that gases have to travel between blood and air. And even though we're talking about very, very tiny distances, it is significant enough to affect the diffusion rates of these gases. And instead of taking only a quarter of a second to equilibrate, it might take up to a half a second or longer. And this can begin to cause problems because, remember, the red blood cells only spend about 3/4 of a second flowing through the alveolar capillaries. And so we start to approach that limit, and that begins to interfere with diffusion rates. Remember also that the degree of perfusion of alveolar capillaries does tend to be affected by how well an alveolus is working. We said in most parts of the body, if oxygen levels drop, we tend to get dilation of a blood vessel to send more blood to the body part that's experiencing hypoxia to help increase oxygen delivery to it. But remember, we said in the pulmonary circulation, everything's the opposite. So if we have hypoxia, local hypoxia, or low oxygen levels, that means an alveolus is not working very well, and we don't want to spend a lot of blood on an alveolus that's not working very well. So in the lungs, hypoxia means a dysfunctional alveolus, and so it results in vasoconstriction instead of vasodilation, and we actually shunt blood away from that alveolus until the problem is resolved, and we start to ventilate that alveolus adequately again. And this is a diagram of the ventilation perfusion coupling, again showing that if we have reduced oxygen in the blood vessel around the alveolus, that results in vasoconstriction. Again, I want to send less blood to an alveolus that is not working very well. Of course, if I have lots of oxygen in the alveolus, that results in vasodilation. I send more blood to that alveolus because it's clearly doing a good job with gas exchange. Okay, the next topic is gas transport. And in this topic, we're going to explore, once the gas is finished diffusing into and out of the blood, once we have the gases in the blood, how is it then that the gases are carried from one place to another? In what form are the gases carried? Now, we've been talking about the gases diffusing into solution and basically dissolving into the plasma. And you might think that that's how they're then transported, as just dissolved gas in the plasma. But in fact, that's actually not the main mode of transport. That's just how it begins. The gases diffuse into solution in the plasma. But after they diffuse into solution, both carbon dioxide and oxygen have other means by which they are actually transported. So remember, in the case of iron, when we talked about iron, how free iron is toxic, and we don't tend to transport it freely through the blood. Because of that, we tend to bind it up with transport proteins. We have a similar thing here, that the oxygen and the carbon dioxide tend not to be transported as free gases just dissolved in plasma, but once they make it into the plasma, they then almost immediately become complex to something else, which then does most of the job of transport. So in terms of where we are in the objectives, we are now at objective 155. Describe what is meant by gas transport, which I just did. How are we going to transport the gases through the blood? So first we're going to talk about oxygen transport, and then we're going to talk about carbon dioxide transport. So beginning with oxygen transport, what we see is that most of the oxygen is transported in the blood as bound to hemoglobin. Just like iron binds to a carrier protein called ferritin, oxygen is carried in the blood bound to a protein called hemoglobin. Some of it is transported just in its dissolved state, but you can see that's a very small percentage. Only about 1.5% is actually transported as dissolved in plasma. 98.5% of it is bound to hemoglobin. So what happens is, in the alveolus, as the oxygen dissolves into solution, according to its concentration gradient by diffusion, as it moves into the plasma, from the plasma, most of it almost immediately then binds to hemoglobin inside the erythrocytes. And again, only about 1.5% remains dissolved in the plasma. So the blood actually contains a lot more oxygen than you would think just by looking at the plasma. Again, the plasma is going to have a PO2 of about 95. That's oxygenated blood. That's this 1.5% of the oxygen that's creating a plasma with a PO2 of 95 or so. Again, much more oxygen is actually held up inside the erythrocytes in the hemoglobin. All right, so we want to talk a little bit about hemoglobin at this point. We've already talked about it a little bit in the first chapter, the blood chapter, but we're going to take a slightly closer look at it now. Excuse me. Recall that hemoglobin is a quaternary protein. It consists of 4 tertiary subunits called globin, and each globin chain contains a heme group at its center, and each heme group contains an iron, and that iron can bind to 1 molecule of oxygen. So one hemoglobin molecule potentially can carry up to 4 oxygen molecules. Something we mentioned when we first talked about hemoglobin is that it doesn't always have to carry 4 molecules of oxygen. Sometimes it can carry only 3, sometimes 2, sometimes 1. So it exists in different states of what we call saturation. And so a hemoglobin molecule that contains 4 oxygen molecules, we say, is 100% saturated with oxygen. If it's carrying 3 oxygen molecules, then the hemoglobin molecule is said to be 75% saturated. Of course, if it has only 2, that's half of its capacity. It's 50% saturated, and so on. So hemoglobin can exist in various states of saturation. Hemoglobin that is bound to oxygen is referred to as oxyhemoglobin. And again, it doesn't matter how saturated. It can be 25%, 50, 75%, or 100. It is still referred to as oxyhemoglobin. So oxyhemoglobin exists in different saturation states. Hemoglobin that has no oxygen bound to it is referred to as deoxyhemoglobin. Deoxyhemoglobin, and often abbreviated as HHB. Where's this extra H come from? As we'll see when we talk about carbon dioxide transport, deoxyhemoglobin can function as a buffer, and it's very good at picking up hydrogen ions. So the HHB refers not to the fact that it doesn't have oxygen on it, but it reminds us that deoxyhemoglobin actually functions as a pH buffer because it can pick up excess hydrogen ions, which of course determine the pH of an environment. To understand how hemoglobin behaves in terms of picking up oxygen and releasing oxygen, we typically explore that by looking at something called the oxyhemoglobin dissociation curve. Oxyhemoglobin dissociation curve. So this is objective 160. The oxyhemoglobin dissociation curve is going to be the foundation of how we understand hemoglobin to behave and how oxygen is transported through the blood. So let's look at our baseline to begin with. First of all, it's a graph, and we need to understand what the axes of the graph are always to appreciate the graph. On the x-axis, we have the partial pressure of oxygen of the blood. This x-axis represents the partial pressure of oxygen in the blood. All right. On the y-axis, we have the percent saturation of the hemoglobin. And remember, hemoglobin can range from 0% saturation, where it has no oxygen bound to it, essentially it's deoxyhemoglobin, all the way up to 100% saturated, where all the hemoglobin has where the hemoglobin has 4 molecules of oxygen bound to it. So on this y-axis, we're talking about the percent saturation of all the hemoglobin in the blood. We're not looking at a single hemoglobin molecule. We're evaluating what is the average state of saturation for all the hemoglobin in the blood. So if you were looking at a single hemoglobin molecule, you'd only have 4 possibilities here. You'd have 0, 25%, 50%, 75%, and 100%. Those are your only possibilities. I think that was 5 possibilities. But notice we have a range from 0 to 100 because we're averaging the percent saturation of all the hemoglobin molecules in the bloodstream. All right, so what do we see? Clearly, this is not a constant. Clearly, these are variable numbers. So first of all, let's appreciate on the x-axis what we're talking about here. Again, partial pressure of oxygen in the blood. Remember that as as the blood flows through the systemic and pulmonary circuit, its partial pressure changes. Recall that we said that the typical PO2 of oxygenated blood was about 94, right? 95, somewhere up in here. And the partial pressure of the tissues, of the blood in the tissues, was about 40. That's deoxygenated blood coming out of the tissues. So we expect typically that the PO2 of the blood is going to fluctuate between, let's say, almost 100, just for the sake of making the numbers even, rounding off, to down around 40. So oxygenated blood, deoxygenated blood, oxygenated blood, deoxygenated blood. And it's going to go back and forth as we go through the pulmonary and systemic circuit. So as we do this, we want to notice what happens to the hemoglobin molecules as we go from oxygenated to deoxygenated blood. Well, in the lungs, in oxygenated blood, we would like the hemoglobin to be primarily loaded with oxygen. I mean, the goal again in the alveolus is to pick up oxygen. And so in oxygenated blood that we see around the alveolus or coming out of the alveolus, we would like most, if not all, of the hemoglobin molecules to be fully loaded with oxygen. And so in these conditions, up near 100 by the alveolus, what do we notice? Excuse me. The percent saturation for the hemoglobin is about 100%. So if things are working normally, about 100% of the hemoglobin that comes out of the lungs in oxygenated blood should be fully saturated with hemoglobin. Then we head down towards the tissues and the systemic circuit. And now we find ourselves in environments where the blood is down to 40, a PO2 of 40, because we're starting to lose oxygen to the tissues. Well, notice when we're in an environment where the blood has a PO2 of 40, the percent saturation on average of the hemoglobin is about 78%. About 78%. So what we're seeing here is that the PO2 of the plasma, in which the erythrocytes are bathed, the PO2 of the environment, in other words, affects the affinity of hemoglobin for oxygen. I'll say that again. The PO2 of the environment affects the affinity of hemoglobin for oxygen. When I say affinity of hemoglobin for oxygen, I mean the degree to which hemoglobin tends to bind to oxygen. If the affinity is high, that means the hemoglobin really likes to strongly bind to oxygen. If the affinity of hemoglobin for oxygen is lower, then it doesn't bind so strongly to oxygen. And that means you'll have less of the hemoglobin molecules being fully saturated. How does this happen? Again, this is all due to structural changes in the tertiary subunits of hemoglobin. Remember, proteins with tertiary structure are held together by relatively weak kinds of bonds. Their three-dimensional structure is formed by things like hydrogen bonding and disulfide bonding. These are weak bonds that can easily be changed. And in the case of hemoglobin, one of the things that alters the shape of the globin is the PO2 of the environment. And it alters the globin in such a way as to reduce the hemoglobin molecule's tendency to bind to oxygen. It doesn't eliminate it, it just reduces it. So in high PO2 levels, the hemoglobin molecule behaves just the way we want it to. It has a high affinity for oxygen, and it really loads up and picks up the oxygen when the blood is in the lungs. Just what we want it to do, getting, again, virtually 100% saturated. But out at the tissues, in that lower PO2, the hemoglobin destabilizes a little bit and releases some of its oxygen. Which, again, if you think about it, is exactly what we want hemoglobin to do. We want the hemoglobin to at least release some of its oxygen as the blood flows through the tissues. This is the delivery of oxygen to the tissues. We don't want the hemoglobin to have as much affinity. We don't want it to hog oxygen to itself. We want it to start releasing it to the tissues. And that's what it does. I think the thing that surprises most people about this oxyhemoglobin dissociation curve is that only about 22% of the oxygen that the hemoglobin molecules contain is actually released to the tissues. Right? So when we get to a PO2 of 40, we only destabilize the hemoglobin molecule enough that it releases on average about 22% of the oxygen that it contained. What this means is that deoxygenated blood with a PO2 of 40, which is deoxygenated blood, is actually not deoxygenated at all. In deoxygenated blood, the hemoglobin is still 78% saturated with oxygen. There's still a lot of oxygen in that hemoglobin. Now you might say to yourself, well, why does the molecule behave this way? Why doesn't it release 30% or 40% or 50% of its oxygen when it arrives at the tissues? Well, there's a couple answers to this. One is it doesn't need to. As we know just from experience, this 22% of gas is plenty to support the metabolic activity of most tissues at rest. We don't need any more oxygen than that. The second reason is a strategic one. We want to have a reserve of oxygen to dip into. We want to have a lot of extra oxygen on the hemoglobin molecules in deoxygenated blood in case we need it. Right? We don't know, for instance, if something's going to happen and we're going to stop breathing for a few moments, a few minutes maybe. So if we stop breathing and we can't get this oxygen that we normally get at the alveolus, and the blood can't get back up to 100%, notice even deoxygenated blood still has a certain amount of oxygen in it that we can dip into if we have to sustain life for those precious minutes until we can be revived, perhaps, say, by CPR. So we don't need more than 22% is one kind of answer to that question. And a second kind of answer to that question is we want to have a reserve that we can dip into. And this creates the very beneficial shape of this oxyhemoglobin curve. Notice it's not a straight line. It's not a straight line. It's what we call a sigmoid curve, an S-shaped curve. And this S shape to this curve is very important. It's very significant, very beneficial in terms of how the molecule functions. And so in objective 161, we're asked to talk about why this S shape is beneficial in terms of the adaptive behavior of hemoglobin. So let's talk a little bit about, again, why this S shape is beneficial. First of all, again, we only need about 22% of the oxygen that hemoglobin carries. If this was a straight line, visualize this, if it was a straight line, then when we get down to a PO2 of 40, we'd be down right about here at about, 55% saturation, somewhere in this ballpark. Well, again, that would mean that the hemoglobin would always be delivering about 45% of its oxygen under normal circumstances. Again, we simply don't need that much oxygen. So if it was a straight line, it would be delivering too much oxygen by the time the blood arrives to the tissues. So we need it to be a little flattened here at this upper end of the curve. We don't want it to dip down too quickly. Another reason we don't want the curve to dip down too quickly has to do with this upper portion right here. Notice that it's extremely flat in these upper values of PO2. Between 108, between a PO2 of 100 and a PO2 of 80, it hardly changes at all. So even if the PO2 is about 80, we still get almost 100% saturation of the hemoglobin. This is extremely important because remember, these numbers that we're using are all numbers that are at sea level, assuming a total atmospheric pressure of 760. So remember what we said about a place like Albuquerque at higher elevation. The atmospheric pressure in Albuquerque is in the lower 600s. And so what that means is that the PO2 of the air in the alveolus in Albuquerque is not going to be close to 100 after equilibration, because we're starting with a lower PO2 in the air to begin with. So in Albuquerque, the PO2 in the lungs of the blood, the best you're going to accomplish might only be somewhere around 90 or even less, depending. And again, as you go up higher and higher and higher in elevation, the PO2 of alveolar air is going to get lower and lower and lower. Remember what we said earlier. This is not due to a change in the concentration of oxygen. This is due to a reduction in the overall pressure of the atmosphere. So as the overall atmospheric pressure goes down, the partial pressure of all the gases in that gas mixture go down as well. So in a place like Albuquerque, the best we might be able to do in terms of oxygenation in the alveolus is, let's say, a PO2 of 90. But notice, because the curve is so flat at this upper end, even at a PO2 of 90 in a place like Albuquerque, we're still able to get almost 100% saturation of hemoglobin. So this extremely flat part of the curve is very adaptive, and it's what allows humans to live at a variety of elevations. Right? Again, if the curve dropped down as a straight line, then by the time we got to 90, we'd already be almost down to only 90% saturation of the hemoglobin. And that's a significant loss of oxygenation. And we don't want that to happen just because we're living a mile above sea level. The other adaptive portion of this curve is this steep portion right here. So notice, once we pass a PO2 of 40, we enter this steep portion where we really start unloading oxygen rapidly from the hemoglobin molecule. So we have to ask ourselves, well, why is this portion so steep, which creates, by the way, the S shape to this, the flat upper part and then the steep middle part. Why so steep in this area? Well, let's think about when the body is likely to experience these kinds of PO2s. There are a variety of situations where you might experience this, but I think the one that makes the most intuitive sense is exercise. During exercise, blood flowing through your muscles, for instance, will experience dramatic reductions in the PO2 because exercising muscle increases its metabolic rate tremendously, and it begins soaking up oxygen quite a bit from the blood to fuel muscle contraction and aerobic respiration that supports muscle contraction. So during exercise, we see the PO2 drop quite a bit in the blood that's in the blood that serves the skeletal muscles. So it wouldn't be uncommon for the PO2 of that blood to drop down to 20. What would we like the blood to be able to do under these exercising conditions? Well, during exercise, we're going to need more than just this normal 22% delivery rate of oxygen. We're going to need a lot more. And so what happens is, at this point, just when we need the hemoglobin to really start delivering more oxygen, we see that it destabilizes very quickly and starts very rapidly releasing its oxygen content. And so the percent saturation from 40 to 20 drops by quite a bit. It drops from 78% down to, let's call it 30%. Right? And so you get a release from 78 to 30, you get a release of 48% of the hemoglobin's oxygen in that short range from 40 to 20. Whereas before, from 100 all the way down to 40, we only released 22% of its oxygen. So we get this rapid release of oxygen from the hemoglobin exactly when we want the hemoglobin to start unloading its oxygen very rapidly, when the skeletal muscles need that extra oxygen. All right, and so again, it's good that we have this reserve of oxygen in the hemoglobin that we can tap into during times of extra need, again, such as exercise. So everything about the shape of this curve, this sigmoid shape of the oxyhemoglobin dissociation curve, shows us how beautifully the hemoglobin molecule is designed to do its job. Pick up oxygen in the lungs, release about 22% of it under normal conditions to the tissues, and during exercise, increase the rate at which it releases the oxygen to fuel contracting muscle cells. Okay, so that's oxygen transport, again, mostly as hemoglobin, mostly in hemoglobin. Carbon dioxide transport, we're starting here with now objective 162 on carbon dioxide transport. Carbon dioxide transport is a little more complicated. Carbon dioxide actually has three different ways in which it is transported. And these three different modes of transport for carbon dioxide are carbonic acid, carbamino compounds, which essentially is carbaminohemoglobin. Recall that hemoglobin can also bind to carbon dioxide, and hemoglobin bound to carbon dioxide is called carbaminohemoglobin. Again, do not confuse this with carboxyhemoglobin. Carboxyhemoglobin is carbon dioxide bound to carbon monoxide, and that's of course very dangerous because carbon monoxide competes with oxygen for the same binding site on the heme group and is about 100 times more, hemoglobin has about 100 times greater affinity for carbon monoxide than it does for oxygen. So you can suffocate due to carbon monoxide poisoning, forming carboxyhemoglobin, which just simply won't release the carbon monoxide and you can't get oxygenation. But with carbon dioxide, that's not the case. Carbaminohemoglobin is not permanent. It's transitory. It's temporary, again, as we'll see. And carbon dioxide does not compete with oxygen for the same binding site. So again, some of the carbon dioxide is in the form of carbonic acid, some is in the form of carbaminohemoglobin, and some remains simply dissolved in the blood plasma. Now, of these three transport modes, carbonic acid is by far the most common. About 90% of carbon dioxide is transported in the form of carbonic acid. About 5% is going to be bound to hemoglobin as carbaminohemoglobin. And then, of course, the remaining 5% is going to be simply carbon dioxide that's dissolved in solution, dissolved in the plasma. All right, just to review, when we say carbon dioxide is transported in the form of carbonic acid, we see this chemical equation that I've put up on the board previously several times and told you that it was an important one that you wanted to remember. So here we see it again. Carbon dioxide dissolves into solution, mixes with water, and becomes H2CO3, which is known as carbonic acid. Now, in reality, most of the CO2 doesn't remain as carbonic acid. Most of the CO2, as we're going to see, is most of the carbonic acid then is going to dissociate into HCO3 minus, a anionic compound known as bicarbonate, and a hydrogen ion. Okay, so really most of the carbon dioxide is transported in the form of bicarbonate. So notice, mixed into this bicarbonate molecule is the original CO2, the carbon and two oxygens. All right, so it might be more appropriate to say that it transports as bicarbonate, and in fact I believe most textbooks would say that the carbon dioxide is transported as bicarbonate. Your book chooses to call it bicarbonic acid for some reason, but it ultimately ends up as bicarbonate. Again, another 5% binds to the hemoglobin to form carbaminohemoglobin. It does not bind to the heme group. Carbon dioxide has its own binding site on the globin chains, so it binds to the protein chains of the hemoglobin molecule. There are some other plasma proteins that will bind carbon dioxide as well, but for our purposes, we're going to focus mostly on carbaminohemoglobin. All right, and notice down here, hemoglobin can transport oxygen and carbon dioxide simultaneously. So, as I said earlier, unlike the situation with carbon monoxide, theoretically it's possible for a hemoglobin molecule to have oxygen and carbon dioxide on it at the same time. However, it's not common for the hemoglobin molecule to carry both. Usually, it carries one or the other, and as it turns out, the more oxygen a hemoglobin molecule picks up, the less likely it is to bind to a carbon dioxide. And conversely, the less saturated a hemoglobin molecule is with oxygen, the more likely it is to bind to CO2. So there's kind of a snowball effect in both directions. As it picks up oxygen, it tends to then pick up more oxygen and release CO2. As it releases oxygen, it then tends to release more oxygen. That creates the steep part of the curve and tends to pick up CO2. So theoretically, yes, it can bind to both at the same time, but in practice, usually it tends to have a snowball effect, either pushing it towards full saturation or carbaminohemoglobin at the other end. And again, as we said earlier, 5% or so of the CO2 is in the form of dissolved gases. This just shows that in terms of the amount of CO2 that actually diffuses between blood and alveolar air, the percent of CO2 that actually diffuses does not perfectly match up with the percent of where it's actually being carried in the blood. So even though 95% of it is in the form of bicarbonate, only about 70% of what actually diffuses comes from bicarbonate, and that's because we can never totally exhaust the blood supply of bicarbonate. We always have some bicarbonate in the blood, so we can't exhaust all of that. And conversely, since that goes down, the percent that the others contribute to diffusion goes up. But again, that's just a minor point. We've already talked about carbon monoxide forming carboxyhemoglobin and the threat of carbon monoxide poisoning. I said earlier that the affinity of hemoglobin for carbon monoxide is 100 times greater than for oxygen. Once again, I was wrong on that. It's in the ballpark, but according to your book, it's 210 times the affinity of hemoglobin for carbon monoxide as for oxygen. And so this makes it very difficult to displace the carbon monoxide once it binds to the hemoglobin. And of course, this creates problems for smokers because you have a certain amount of carbon monoxide in cigarette smoke. And notice in heavy smokers, this should be alarming. About 10% of your hemoglobin is carboxyhemoglobin and not available for oxygenation. And so that the danger there speaks for itself. Okay, so in terms of how we pick up or load and unload carbon dioxide, let's take a look at some detail at what happens. Carbon dioxide diffuses into the blood. All carbon dioxide begins by diffusing into the blood, but again, only about 5% remains in that state. 95% of that carbon dioxide will enter the red blood cell and go through this reaction that we said, forming carbonic acid and then bicarbonate. Now, why does this reaction happen mostly inside the red blood cells? Well, it happens mostly inside the red blood cells because there's an enzyme inside the red blood cells that facilitate or catalyze this reaction. And that enzyme is known as carbonic anhydrase. So while this reaction occurs a little bit out in the plasma, the vast majority of it happens inside the erythrocyte because of the enzyme carbonic anhydrase. And so we get the buildup of carbonic acid inside the red blood cell. But of course, if we just kept it as carbonic acid, that would be a problem because the inside of the red blood cell then would become extremely acidic as the carbonic acid dissociates into bicarbonate and hydrogen ions. All these hydrogen ions would create a major problem for the erythrocyte interior. One of the things it would do is, for instance, this very low pH would damage the hemoglobin itself. So what do we do with this hydrogen? And what do we do with this bicarbonate? Because remember, bicarbonate is a buffer. It can either pick up hydrogen ions or release yet another hydrogen ion depending on the conditions. And any molecule that can pick up or release hydrogen ions can function as a buffer. Well, as it turns out, bicarbonate is such a valuable buffer that we don't just leave it inside the red blood cell. We want to make use of it, at least temporarily, and so what we do is we send it out to the blood plasma. This bicarbonate does not remain inside the red blood cell. We send it out to the blood plasma where it functions as a buffer. Now remember, again, something we talked about way back in the first chapter of this semester, blood has a very narrow range of pH. The pH of the blood has to be slightly alkaline, somewhere between 7.35 and 7.45. There's a couple of mechanisms for maintaining that very narrow range of pH. Respiratory rate, which affects carbon dioxide levels, the kidneys are involved in regulating pH, but this bicarbonate is a principal factor that helps keep the blood in that very narrow pH range, the blood plasma in that very narrow range of 7.35 to 7.45. So this bicarbonate, which again contains the carbon dioxide that we're carrying, we put it to good use. In other words, we put it out into the plasma where it helps buffer the blood. Well, if I'm going to pump these bicarbonate molecules out of the red blood cell, notice what's going to happen. I'm going to be pumping out or extruding from the cell a negatively charged entity. Well, that's going to disrupt the membrane potential, right? If I take all these bicarbonate molecules, which there are a lot of, because remember 95% of your CO2 is becoming this bicarbonate molecule. Well, if I'm pumping those out of the cell, I'm losing a ton of negative charge, and that's gonna mess up the membrane potential. Well, we can't allow that. So, as it turns out, as we pump the bicarbonate ions out, we also pump something else in to replace it. So we have essentially an antiport, a carrier molecule that pumps two separate molecules or entities in opposite directions. What do we exchange for the bicarbonate? Well, we exchange something else that's got a negative charge. So in other words, we're trying to replace the negative charge that we're losing, and that ends up being the chloride ions. So for every bicarbonate that gets pumped out of the erythrocyte, we replace it with a chloride ion that we're bringing in from the plasma. The chloride ions really don't have any physiological effect on the red blood cells. They're essentially neutral. They don't affect the red blood cell in any way, but what they do is they maintain the membrane potential because they're replacing the negative charge that we're losing when we pump out the bicarbonates. This is all referred to as the chloride shift. The chloride shift is replacing the bicarbonates that the erythrocyte loses with the chloride ions that we're pumping in at the same time. But again, so that explains why the loss of the bicarbonate doesn't upset the membrane potential. But again, what about these hydrogen ions that are building up inside the erythrocyte? What do we do with those, and how do we prevent them from damaging the erythrocyte? Well, you'll notice this last bullet here, these hydrogen ions that are building up, they bind to hemoglobin. And you might recall earlier in this presentation, I talked about or the PowerPoint showed that one way to represent deoxyhemoglobin, hemoglobin that has no oxygen on it, was to refer to it as HHB. And I mentioned earlier why HHB. So this is hemoglobin, right? HB. Why the HHB? Well, because deoxyhemoglobin, as it turns out, can function as a buffer. So we see, again, that this hemoglobin is a marvelously designed molecule. Not only is it perfectly designed to load and unload oxygen, as we saw with the oxyhemoglobin dissociation curve, but now we see that when it's deoxyhemoglobin, it can function as a buffer that can bind to hydrogen ions and become HHB. This is what prevents the hydrogen ions that are building up here as we're sending 95% of our carbon dioxide through this reaction. These hydrogen ions are binding to the deoxyhemoglobin to become HHB, deoxyhemoglobin. Another way of referring to that, by the way, is also as reduced hemoglobin. All right. Now, the hydrogen ions that are initially building up also do something amazing, and that is the pH change, the acidic conditions, actually destabilize hemoglobin and reduce its affinity for oxygen. So one of the reasons why hemoglobin starts releasing its 22% of its oxygen when it arrives at the tissues is because the increase in CO2 causes an increase in hydrogen ions inside the cell, inside the erythrocyte, and that is what destabilizes the hemoglobin and starts causing it to release its 22% of its oxygen. And so as these hemoglobin molecules release the oxygen, of course, they become the deoxyhemoglobin that is then very good at picking up these hydrogen ions and buffering the internal status of the erythrocyte. All right, now this is all probably best understood by looking at a diagram. And so here we see a diagram of what's happening with both oxygen and carbon dioxide in the tissues. Okay, so here we're not talking about in the lungs. Here we're talking about what's going on in the tissues. Again, notice in the tissues we have a high level of CO2 because we are respiring and producing CO2 as a waste product. So we've got to do something with this excess CO2. Remember, in terms of the amount that actually diffuses, it's 7% diffusing from the tissues and then ending up as dissolved in the carbon dioxide. 23% of the diffusing carbon dioxide ends up as carbaminohemoglobin, carbon dioxide bound to hemoglobin, and 70% of the diffusing carbon dioxide ends up going through this reaction inside the red blood cell to produce carbonic acid and then bicarbonate. And again, it's carbonic anhydrase, the enzyme carbonic anhydrase, that's required for this reaction to proceed effectively. All right, now again, these percentages reflect how much of the diffusing carbon dioxide goes through these channels. Once we get out here, if you were to look at the sum total of all the carbon dioxide that's being carried in the blood, it's 5%, 5%, and 95%. All right, as we've been saying, the carbonic acid dissociates into bicarbonate and hydrogen ions. As I mentioned earlier, we take most of the bicarbonate and pump them out into the plasma to serve as a buffer, but at the same time, we pump chloride ions in to maintain the electrical balance, the membrane potential. So that's an antiport molecule, an antiport pump. It's pumping two separate things at the same time, and this is known as, again, the chloride shift. What happens to these hydrogen ions? Well, again, the hydrogen ions are destabilizing the oxyhemoglobin. The pH change caused by these hydrogen ions is destabilizing the oxygen, the oxyhemoglobin, causing at least some of it to give up its oxygen and become deoxyhemoglobin. And there's the oxygen that's been released, which is perfect because where are we? We're in the tissues. And we would like the hemoglobin to start releasing some of its oxygen. So this oxygen then diffuses along its concentration gradient out to the tissues. At the same time, recall a very small percent of the oxygen that's dissolved in the plasma is also going to be diffusing along its concentration gradient. And so this is again, the 22% or so of the oxygen content of the hemoglobin that typically diffuses out into the tissues. And so the deoxyhemoglobin that's produced by this does what? Well, it picks up these hydrogen ions so they don't continue to build up and cause extreme pHs that damage the cell. And the deoxyhemoglobin becomes the so-called HHB, right? Deoxyhemoglobin or reduced hemoglobin. Again, that serves as the internal buffer. So this is a very nice diagram that illustrates everything we've been talking about. As we move into the alveolus, of course, what we're going to see is that everything reverses. And so this diagram is an illustration of what happens when the blood arrives at the alveolus. So now instead of tissues out here, we have alveolar air. And remember, the concentration gradients completely flip-flop. And so now we have less CO2 out here, more O2. And so essentially everything that we've been talking about is driven in reverse. The high oxygen levels cause diffusion in. Those high oxygen levels move in. And remember what we said, once hemoglobin starts picking up oxygen, it tends to be a snowball effect, and it'll pick up more and more oxygen and convert from deoxyhemoglobin to oxyhemoglobin. Well, as it goes through that conversion to oxyhemoglobin, it releases its hydrogen ion, and that hydrogen ion is now available, and the chloride shift reverses under these conditions, and bicarbonate starts being pumped back into the erythrocyte, and chloride is pumped out. So now I start getting high levels of bicarbonate and hydrogen ions present from these two reversals of these processes. And that high concentration of these things starts to drive the reaction back in the opposite direction. Remember, most chemical reactions are reversible, even enzyme catalyzed ones. And reversible reactions are pretty much driven by the concentration gradients of the reactants and the products. So this simple increase in concentration drives the reaction back towards carbonic acid, which then drives the reaction back to water and carbon dioxide, and that carbon dioxide then is released and diffused along its concentration gradient into alveolar air. We also see that the carbamino hemoglobin that's bound to carbon dioxide starts to destabilize under these conditions, and the hemoglobin releases its carbon dioxide, which then again is free to diffuse into the alveolar air. And of course, the dissolved CO2, that small percentage that's in the plasma, also moves along its concentration gradient out into the alveolar air. And so again, everything we want to happen happens in this environment of the alveolus. The blood unloads carbon dioxide and loads oxygen. Okay, so that pretty much takes us up to approximately objective number 170, 171, somewhere in that ballpark. So what we're going to do next time is we're going to see if there are any adjustments that can be made to the behavior of hemoglobin that will affect its behavior. Are there any things that we can do, or anything that happens to the hemoglobin molecule that can alter its affinity and make it behave slightly differently under different conditions? And we're going to begin that discussion somewhere around objective number 176. So looking at it carefully, it looks to me like we've made it up through objective number 175, and we will pick up next time with objective 176, looking at environmental factors that affect the behavior of hemoglobin. Until then, stay safe, and we'll see you next time.